Good morning. My name is Kristen Pluchino, and I'm a clinical reviewer in the Division of Diabetes, Lipid Disorders, and Obesity in the Center for Drug Evaluation and Research at the U.S. Food and Drug Administration. Before getting started, I'd first like to thank c-path for their efforts in organizing this symposium and the numerous speakers and attendees for providing their expertise on this important topic. Today, I would like to discuss aspects of the FDA's regulatory framework that are relevant to the design of new onset type 1 diabetes intervention trials, including the potential use of C-peptide as an efficacy endpoint to support regulatory decision-making. Please note the views expressed in this presentation are mine and should not be interpreted as those of the FDA. As discussed by Dr. Irony, the agency recognizes the unmet need for drug or biologic products that can be used as early interventions in new onset type 1 diabetes. As type 1 diabetes is characterized by progressive loss of beta cell function resulting in absolute insulin deficiency, therapies intending to either preserve remaining beta cell function or attenuate the decline in beta cell function may be potentially promising therapeutics to alter the course of the disease. There are currently no drug therapies approved for this clinical use, and therefore no precedent for the regulatory framework that may be used to approve such therapies and make them available for patients. Generally speaking, for a drug or biologic product to be approved for marketing in the United States, FDA must determine that the drug is effective and that its expected benefits outweigh its risks. Clinical benefit is defined as a positive, clinically meaningful effect of an intervention on how a patient feels, functions, or survives, and efficacy endpoints are used to assess the benefit of medical products in clinical trials. There are two main types of efficacy endpoints. Clinical endpoints directly measure how a patient feels, functions, or survives. In other words, clinical endpoints directly measure clinical benefit. For example, the incidence of severe hypoglycemia, DKA, or mortality would all be considered clinical endpoints as they are direct measures of meaningful events that are important to patients. In contrast, surrogate endpoints are used as a substitute for a direct measure of how a patient feels, functions, or survives. Surrogate endpoints are often based on the measurement of a biomarker, such as a laboratory measurement. A surrogate endpoint does not directly measure a clinical benefit of primary interest, but is rather expected to predict a benefit. There are three main types of surrogate endpoints which can be characterized by the level of clinical evidence supporting their use. Surrogate endpoints that have undergone extensive evaluation are called validated surrogate endpoints. These endpoints are accepted by the FDA as evidence of benefit and can be used in lieu of a clinical endpoint for traditional FDA approval. HbA1c is an example of a validated surrogate endpoint. Hence, HbA1c can stand in for the clinical outcome of microvascular complications in a clinical trial. At the opposite end of the spectrum, endpoints that have the least amount of evidence supporting their use are termed candidate surrogate endpoints. While these endpoints don't support regulatory decision-making, they can be useful in various contexts, such as when used in proof of concept or hypothesis-generating studies. Intermediately, a surrogate endpoint for which strong mechanistic and/or epidemiologic evidence supports that an effect on the surrogate is expected to be associated with a clinical benefit, but without sufficient clinical data to validate the surrogate endpoint, is considered a reasonably likely surrogate endpoint. Reasonably likely surrogate endpoints can be used in FDA's accelerated approval pathway. The role of C-peptide as a biomarker has not been established in the regulatory setting, but its use as a reasonably likely surrogate endpoint may be justified. Note that even if we think an endpoint is reasonably likely to predict clinical benefit, surrogate endpoints for which the clinical benefit cannot be quantified present significant challenges in the FDA's regulatory review process. Consider a hypothetical drug therapy for new onset type 1 diabetes that shows an increase in C-peptide in a clinical trial. However, some serious risks are also identified in the trial. How do we decide if the trade-off for those risks are acceptable if we cannot correlate the measured change in C-peptide with an actual clinical benefit that is important to patients? The type of surrogate endpoint also has an impact on the options for regulatory approval. Traditional approval is based on a clinical endpoint or a validated surrogate endpoint, and therefore the ability of the treatment to provide benefit to patients is generally well characterized at the time of FDA approval. Most FDA-approved drugs are approved via the traditional approval pathway. In contrast, the accelerated approval pathway can allow for approval of a treatment based on a surrogate endpoint that has not been validated but is reasonably likely to predict a clinical benefit. The accelerated approval pathway is intended to provide patients with serious conditions, such as type 1 diabetes, more rapid access to promising therapies. In this program, a drug may be granted approval based on a reasonably likely surrogate endpoint, with the commitment of the pharmaceutical company to perform post-marketing studies to confirm the anticipated clinical benefit. In the case that the confirmatory trial does not show that the drug provides clinical benefit, the FDA has regulatory procedures in place that could lead to removal of the drug from the market. The main benefit of accelerated approval is that it allows for faster access of promising treatments to patients with serious conditions. The principal downside of this approach is the possibility that patients will be exposed to risks from a drug that ultimately will not be shown to provide an actual clinical benefit. In addition, with regards to the safety assessment, there generally will be fewer, smaller, or shorter clinical trials than is typical for a drug receiving traditional approval. There's also the concern that the confirmatory trials may not be completed in a timely fashion. C-peptide is currently being used as an efficacy endpoint in new onset intervention trials. C-peptide is secreted in equimolar amounts to insulin. Therefore, it can be considered a biomarker of beta cell function and endogenous insulin secretion and is considered a surrogate endpoint. In general, the type of surrogate will impact the clinical development program. In the case of C-peptide, there may not be sufficient evidence at this time to support its use as a validated surrogate endpoint. Therefore, let's consider a scenario in which C-peptide is used as a reasonably likely surrogate endpoint in a new onset intervention trial to support accelerated approval. Questions we may need to answer in order to take this approach are: what are the anticipated clinical benefits of preserving C-peptide in the post-marketing trial? For example, better glycemic control, less hypoglycemia. Is reduction in insulin dose, even with the same number of daily injections, meaningful to patients? Additionally, what amount of C-peptide preservation would be indicative of a meaningful impact on the clinical benefit of interest? Is this amount different between populations? For example, adults versus pediatrics? Note, just because a result is statistically significant does not necessarily mean that the result is clinically significant. And there should be evidence to support that the magnitude of C-peptide preservation is predictive of a meaningful impact on a clinical benefit. Additionally, what would the confirmatory study look like? For example, how long should subjects be followed to assess long-term benefit of the drug, especially when a drug may have the potential for serious harm? What is the most endpoint to confirm the anticipated clinical benefit? Also, if clinical benefit is perceived based on the accelerated approval of a product, to what extent does this raise challenges for the ability to recruit patients into the required post-marketing study if it is placebo-controlled? Furthermore, as my colleague Dr. Donna Schneider will discuss tomorrow, the ethics of continuing placebo-controlled arms should be considered, particularly if equipoise between treatment arms becomes questionable. I want to make the additional point that the accurate measurement of a surrogate endpoint is fundamental to ensure confidence in the results from a clinical trial. There are various C-peptide assays currently in use which may have different performance characteristics, such as the accuracy and precision of an assay. Careful consideration should be given to whether the performance characteristics of a given C-peptide assay are adequate to support regulatory decision-making, and also to be aware of the assays used when interpreting clinical trial results. To summarize, FDA is aware of the unmet need for disease-modifying drug therapies in new-onset type 1 diabetes. To approve new treatments, FDA must determine that the expected benefits of a drug outweigh its risks. Benefit is established by the use of either clinical or surrogate endpoints, and there are several types of surrogate endpoints, which are classified by the level of clinical evidence supporting their use. The type of surrogate endpoint also has an impact on the appropriate regulatory approval pathway. C-peptide is a biomarker of endogenous insulin secretion and therefore a surrogate endpoint that potentially may be able to be used as an efficacy endpoint to support regulatory decision-making. Currently available data suggests that C-peptide may be able to be used as a reasonably likely surrogate endpoint in FDA's accelerated approval pathway. However, there are various considerations that should be explored with this approach. For example, what are the ultimate clinical benefits? What is the magnitude of C-peptide change that would be indicative of a meaningful difference on a clinical benefit? Does this magnitude differ between populations? And various trial design considerations related to the required post-marketing study. We look forward to hearing comments regarding this important topic at the upcoming panel discussion. Thank you for your time and consideration.